Hello, my name is Richard Quack. I'm a Senior Consultant Medical Oncologist at the National Cancer Centre Singapore. Today I'm here with my colleague Patrick Tan to present our paper SETD2 histone modifier loss in aggressive GI stromal tumours, which was recently published in Gut. Gastrointestinal stromal tumour, GIST for short, is the most common mesenchymal tumour of the GI tract. It's also by far the most common single entity of sarcoma. Incidence-wise, it's about 15 cases per million population, and the disease is characterized by gain-of-function mutations in KIT or platelet-derived growth factor alpha genes. So since the early 2000s, imatinib has been approved for use in first-line setting metastatic GIST, and it's able to induce high remission rates and prolonged disease control in the setting of metastatic GIST. While in the setting of localized disease, surgery remains the mainstay of treatment. However, despite best optimal surgery, 40% of patients still do relapse. So investigators have been trying to risk predict who are the patients at risk of relapse following surgery and those who are not at risk of relapse. And so there are many clinical parameters or clinical stratification models that we commonly use. Models typically involve the features of the tumor size, the tumor mitosis or mitotic rate, as well as the location of the tumor, with small bowel lesions being worse than gastric lesions. And fourthly, the presence or absence of tumor rupture. While these parameters are very useful for us in the clinics to risk stratify and to predict risk of relapse for our patients, it clearly doesn't give us any insights into the molecular aggressiveness of GI stromal tumors. So we sought to understand what exactly makes GIST malignant.
In order to gain insights into the genetics of high-risk GIST, we sequenced the exomes of a cohort of GIST tumors, comparing mutations found in high-risk GIST tumors compared to low-risk GIST tumors. The initial discovery cohort was 18 cases, but any genes that we found to be recurrently mutated were then taken for prevalence sequencing in an expanded cohort of 120 cases. What we found was that we confirmed that c-KIT and PDGFRA are indeed the most frequently mutated genes in GIST. However, we also found a new gene called SETD2 that was also mutated specifically in about 11% of high-risk GIST. In a series of related experiments, we sequenced the exomes of multiple GISTs isolated from the same patient. What we found was that common driver genes like c-KIT and PDGFRA were mutated in all GISTs obtained from the same patient. In contrast, genes like SETD2 and p53 were often mutated in only certain sites from the same patient but not in others, consistent with genes like SETD2 playing a role in tumor progression. SETD2 encodes a protein involved in the trimethylation of histone H3 at lysine 36, commonly referred to as H3K36Me3. This refers to a particular chromatid modification mark in the human genome that highlights actively transcribed regions. Subsequent characterization of SETD2 by our team suggests that SETD2 is a novel tumor suppressor in high-risk GISTs. First, we found that tumors with SETD2 mutations indeed exhibited lower SETD2 protein expression. These tumors also exhibited genome-wide DNA hypomethylation specifically associated with heterochromatic regions. At the functional level, we were able to silence SETD2 in GIST cell lines, and we discovered that upon SETD2 silencing, these cell lines exhibited increased genomic instability, DNA damage, and sensitization to treatment.
In conclusion, SETD2 is a novel tumor suppressor gene in GIST. These results are clearly very exciting for us clinicians and researchers alike. Importantly, the next step will be to see how we can incorporate SETD2 mutation results into the clinical risk stratification models in common use to allow us to better refine our models and to better risk predict our patients.